חושב שהוא לא חושב.
הוא לא היה שום דבר למודל.
זה היה מדהים בארץ? תגיד, עדי בארץ או הוא לא בארץ? לא, זה כל חודש לפני. עוד שנייה, כן, רגע. אני אדבר על זה עוד רגע, אבל עוד שנייה. Okay, let's start. So there are two technical issues that can you close the door from behind there? So there are two issues that are technical I need to discuss with you. One is putting the lecture in the Moodle, which I don't know how to do myself, so I need I need the address. I don't know the address, and I did not hear, or I don't know if somebody knows how to do it, then I can do it. Is there anybody who knows how to, what is the link, what is the, no. I guess Adi will know. I'll email Adi today and I'll ask him how to do it, or maybe send him the link, my link, and he can put it to the course link. The other thing is from my understanding, but I may be wrong, from my understanding Adi asked me to change, not to present the papers related to my work this Wednesday, but rather a month from today, if I remember well. So I think so, but in the intermission I will check when is the next time that I should be here. I think it's a month from today, but maybe I made a mistake. If it's this Wednesday, then it's a problem, but I remember he told me that there is some change, and he changed the date several times, so if I remember well, it's not this Wednesday. But
you mean our paper will not read the other?
Yes, because for every section, no, every section, let's say my three presentation has to follow with 3 papers to be presented by you around my topic. You meant something else. Oh, what did you mean? It's not on Wednesday.
Wednesday is the paper that we're not presenting, that somebody else that
we have to read it and discuss it with Adi.
So why did you ask me about the paper?
No, because you didn't know if it's your paper or Adi's paper or what's going on.
But now you think it's not my. It's not. I think it's not my paper because Adi told me something much later.
Maybe it's Adi's. I don't know.
So am I wrong or am I right? It's not clear.
You're right.
I'll email Adi both things. I'll email him the link to the presentation and I'll ask him this question after I finish this lecture. Okay, so this is the third presentation and the last one, and so there is a lot of material, so I'll try to do it as I do it in this course, rather shallow. Unfortunately, it's not deep in any sense, in any way, but as I said, my hope is that you will get some new insights, some new excitements, some interesting things that you may have not thought about, and now you may be able to think better about because I presented it. So today I want to speak a little bit, to start to speak about nonlinear dendrites, or to complete the issue of dendrites by talking about the nonlinear dendrites, and then we'll go to the computational part of the dendrite. And if I will have time, I hope I will have time, we'll go further to some of the big projects that are being done today. This is a more general aspect of my talk, not neurons and dendrites, but rather the big projects which you should know about them. You know, the Japanese announced a big project, the American announced a big project, the European announced a big project. So I want to say a few words about this new agenda for brain research. As you will be part of it for sure in one way or another. Okay, so you know that there is a relatively, well, it's not new anymore, it's now 30 years ago, this differential interface contrast video microscope. It came to the field about, I think, 30 years ago. The capability to take a slice, a brain slice, in this case cortical slice, and using optical techniques, differential interface contrast techniques, enable you to see individual cells, which was not the case before, because before people took slice and just impale with the electrode blindly, hoping that the electrode will be will be by chance impaling the cell body or something. Typically the cell body because it's the biggest thing. But now that you have this new technique, now in any lab of course, you are able not only to recognize the cell bodies, but by focusing at different Z depth, you can see also processes. So for example, if you can see there is a cell body here of a pyramidal cell, and you can see the beginning of the apical dendrite going this direction and so on. So this was a big jump, so to speak, in terms of optics, as you understand already. Each time there is a new technique, there is a lot of new things that one can do, including recording visually guided, not by chance, but visually guided if you want this cell to be recorded from this cell or this cell, and of course simultaneously more than one cell, which was also a big jump, because then, actually started by a woman in England, Alex Thompson, who was the first to record from two cells in the cortex, but blindly. This was a heroic thing to be able to record two cells blindly by chance, to get two cells connected to each other via synapse, just to characterize the synaptic connection between cells. But now you can do it easily because you can see the cells, so there is no problem. You can ask whether this cell and these cells are connected and so on. So this is the deep video microscopy, which is, as I said, 30 years ago. The other thing that this deep video microscopy enabled people to do is to do this. So you see, so you know when you are, suppose I want to record from this cell body, let's say, and from the dendrite of this one. But you can see that the dendrite here, it starts here. Can you see what I'm showing? Because the contrast is good enough. Let me just turn it off for 1 second so you can see a little bit better what I'm talking about. So for example, suppose I want to record from this cell body and I want to follow the apical tree far away, let's say here, somewhere there, and record from there. So I need to all the time, adjust the focus in order for me to be able to impale with an electrode here and then also impale with an electrode there. The problem, of course, is that you may, by chance, go to another dendrite. You remember there are many dendrites, one near the other. So, it's very hard to follow. Sometimes you may, let's say, go here, and it will be a dendrite of another cell. So Sackman and Stewart and Sackman, what they did, in order to make sure, using the big video microscopy that I just showed you, to make sure that they do record for the first time ever, basically, from the same cell, both at the cell body, here is the axon, here is the beginning of the basal dendrite, and also from the apical main trunk, because of course there are the side branches, the oblique and other. What they did is they used different stains with an electrode, let's say with green stain, electrode with blue stain, just to see that the stain meet each other. If the stain meets each other, if the dyes meet each other, it must be the same cell. So this was a little trick, and this was the first time that people record. So this is Stewart and Sackman, a very, very famous paper from Nature, in January, Nature 1994. So it's 20 years by now. That they recorded for the first time from the dendrite and from the axons, from the dendrite and from the cell bodies. Soon you will see that also from the axon. And then they found something very, very surprising that you all know. I understood from last time. Just wanted to show you the result, if you didn't see the result. So as you see, you record here, and actually they also succeeded in the same cell to record from the axon of the same cell. So they have 3 recording points simultaneously, 3 points recorded in the cell body, dendrite and axon. And they saw something interesting, that no matter what you do, so for example here you stimulate, you inject current through the dendritic electrode. So you inject current and you start to depolarize. As you can see, you depolarize because this is the electrode that injects the current. So there is a current injection here that you don't see. There is a current injection here through this electrode. You see voltage starts to be developed locally at this injection site. But you see that the action potential, so to speak, starts at the soma, although you inject in the dendrite. So you inject in the dendrite, voltage attenuates from the dendrites to the soma, as we expect. The voltage attenuates from the dendrites to the soma, this attenuation, as you learned already from cable theory. But the action potential actually starts, although the depolarization is bigger here than here, the action potential fires first at the cell body, but actually now that you also have the axon, you see that it starts at the axon. So no matter where I inject the current, if I inject it in the dendrite, if I inject it in the soma, if I inject it in the axon, the action potential tends to start, the sodium action potential, the classical sodium action potential, the black one starts at the axon and then back propagates from the axon to the soma, and then back propagates from the soma to the dendrites in terms of timing. So this is first, second, third. Okay, so this is an interesting result. We knew already from classical experiments that the action potential tends to start not at the soma, although we injected current at the soma, we knew that it tends to start in the axon. We know that the axon is very hot, very excitable, a high density of sodium channels, very high density of sodium channels in the initial segment, but then it back propagates here, and that's what typically people saw for many, many years, the somatic action potential, because they recorded from the soma. But now they also see that there is a big reminiscence, a big trace of the action potential. Big, I mean 50 millivolts in this case, because this is about 100 millivolts, a big action potential going backward. Now the question is, and the question was, and that was the beauty of this work, really very innovative, not only technically, but also in terms of what they wanted to show. The question is whether this backpropagating action potential, so that's the name, backpropagating action potential, BPAP, backpropagating action potential, is it active or passive propagation backward? So we know that if it's passive, if I inject a spike here, suppose I inject a spike here or generate a spike here, there should be some passive propagation backwards. We know that from what we learned. There is a propagation backward. It's a brief phenomenon. So already you know that if it's a brief phenomenon with high frequencies, there should be a steep attenuation backward. As brief as it is, the briefer it is, the more attenuation you would expect. This you already know, and if it's a steady state current here or voltage here, it will attenuate less. This already you know. So the question is this attenuation from here to here, is it passive or active? So what they did, which is really innovative, they did the following. They put TTX in the buff. So they blocked all action potentials that are sodium dependent. Remember, TTX is a very specific dye, already used by Hodgkin-Huxley, to block sodium, voltage-dependent sodium channels, not calcium-dependent or potassium-dependent voltage channels, only sodium-dependent. It's a very specific dye, very, very, very lethal, very dangerous dye, I'm saying, very dangerous drug, very, very dangerous drug with very, very minute, you are completely dead because it blocks your action potential, sodium action potential, it blocks all communication. So that's what they did, they blocked the action potential. So there is no action potential anywhere in the slide, sodium action potential. And then they voltage clamped the soma with the shape of an action potential. Okay, they call it simulated action potential. So they took the recorded action potential from the soma, so this is the recorded action potential from the soma. They had this voltage trace. Now they used the voltage trace recorded experimentally to inject voltage with a voltage clamp, just to really, really draw, so to speak, the same shape of the action potential into the soma, but the soma is completely not active, it's passive, because I blocked all the sodium channels. Is it clear? It's a simulated voltage trace injected to the cell body. Why did they do it? Because they wanted to get the same action potential as they got before the block, and see how much of it reaches this point. Now they know that everything is passive in terms of sodium-dependent voltage channel, and they found, I didn't draw it, but if I would draw it, it would be something like that. Something like that. About quarter of what you see experimentally. So if I inject this to a passive cell that is blocked by TTX, I would get something like that. So this is the backpropagating passive action potential, backpropagating passively. This is this one that I just drew with my finger. But you see that it's much, much bigger. So they realized that there must be some boosting, some help of this action potential to backpropagate via sodium-dependent, voltage-dependent sodium channels. So this was the first time that not only that you saw something big in the dendrite, bigger than expected from passive, but you know that it is being carried, this boosting, being carried by sodium-dependent, voltage-dependent sodium channels in the dendrites. This means somehow that the soma that generates these proteins that become sodium-dependent, voltage-dependent sodium channels, some of them are also going backward into the dendrite. So the dendrite is not passive. Dendrite is excitable because it has these channels. But it doesn't have these channels in high density. Because if it would be a high density, I mean really high density, like in the axon, then there would be fully propagated action potential, full-blown action potential. 100 millivolts here, 100 millivolts. So this will become an axon in some sense. The dendrites will be an axon, but it's not. It does attenuate. It does attenuate, and as you go, now people can record from here and from here also. People like Larkum and Stuart, they can really start to record here using this deep video microscopy and very fine looking and staining at the cell. They can see that this is a backpropagating attenuated action potential. It's not full-blown action potential. It's not healthy action potential. It's half passive, half active. It's not completely passive, but it's not fully healthy. So this is a new phenomena discovered in the brain. We did not know about this phenomena until 1994. We did not know that there is this backpropagating action potential. Certainly we did not know that the dendrites have these active sodium channels and so forth and so on. So this is an interesting thing to know. That's one thing. And of course, and I'm not going to talk about this because this is a whole new thing about the fact that this backpropagating action potential is critical for plastic properties in the dendrites. Critical. So if you activate the synapse here, and at the same time the action potential, or just after that the action potential comes backward to the same location of this excitatory synapse, you know that these synapses undergo LTP. And if it's a reverse on the time and the action potential arrives here before the synapse is active, it will become LTD, the synapse will become weaker. So this signal, this is a critical, important handshaking, we can call it handshaking signal, because it handshakes. It handshakes with the synapse. It tells the synapse, the excitatory synapse. It tells the excitatory synapse, you are activated now. You are part of other synapses, of course, not by yourself, because you are very weak as a synapse. You and your friends together succeeded to generate a spike. In a short period of time, I'm going backward to tell you, I'm sending you a message, I'm handshaking with you, that you are responsible for the spike, so to speak, and so change, become stronger. If this is the order in time. Synapse, action potential. But if not, if there is an action potential being generated by other synapses, so to speak, not by me, and this action potential come to me, but I'm not one of those that generated this action potential, within a given period of time, it means that you are not part of the generation of action potential, become weaker. This is a very important signal, very important message going from, which is a wonderful message if you think about it. It's really wonderful. There is a message, a signal that goes to the synapses and tell them, so to speak, and this was a classical question, by the way, many, many years people realized, how do these synapses know to become stronger or weaker? Because they need to know somehow that there was a success, some feedback. Otherwise, maybe it becomes stronger and weaker without a teacher. So this backpropagating action potential is a teacher, is a supervisor, so to speak. He supervises the synapse and tells the synapse what to do. It's a very critical, important signal, but I'm not going to go into the whole STDP, spike timing dependent plasticity. It's a whole field of research, both theoretical field in physics. What can you learn with these spikes? how much memory can you embed in networks of neurons using this signal within this period of time, classical conditioning and so forth and so on. So this became a new signal, but not only a new signal, but very, very, very important new signal. It's a new spike, it's a new signal that is interacting with the synapses that generate it.
And what happens in inhibitory synapses?
That's a very good question. People are trying to find learning rules, we call it learning rules, STDP, STP, LTP and LTD. And so it seems in general, but of course there are many, many local rules depending on the system that you work in. And you know, for example, backpropagating action potential does not always happen. For example, Purkinje cells do not have backpropagating action potential. So there must be other plastic rules because also there the synapses learn. So there must be other, it's not the only signal, but it's an important signal in the cortex at least. So we don't really have a very good rule. So there is a mathematical rule for this plasticity for excitatory synapses. There is no clear agreed rule on inhibitory synapses in terms of plasticity. It's not so easy to find inhibitory synapse and to show that when this is active and it reduces the chance of an action potential because it's inhibitory, in principle it should go the opposite in terms of timing. So the positive timing should be LTD and the negative timing should be LTP. Some people did show it. But again, I don't want to go really into it too much because the whole direction of computer learning and machine learning is really based on some ideas coming from this supervised and unsupervised learning and so forth. This is a supervised signal, but it's intrinsic supervised because nobody from the outside tells this action potential to generate. It's part of the input system. I just wanted to highlight a progress that was made 20 years ago due to this development of new technologies that enable you for the first time to see locally what happens in dendrites. As I said, this was a completely unknown country, the dendrites. Nobody recorded from them. It was just unknown. You know that synapses are there anatomically, but what are the signals locally? Absolutely unknown until you started to record. Now there are these optical measures and the calcium imaging that I mentioned.
How do they know for sure? I mean, I don't know, but the 75%, approximately 75%, is active backpropagating action potential that comes from the soma to the dendrites, and not that you have something like a closed network that comes back through the dendrites.
That you think maybe this axon somehow goes backward and gives this synaptic input, So as I said, first of all, when you block with TTX, there are no action potentials, so no synaptic activity.
But then they got 25%.
Right.
But
so the additional one.
Yes.
You ask whether the additional one comes from synapses. Okay, so first of all, it could be in principle, but we know two things. First of all, that a single axon, a single action potential, a single synapse, does not generate a spike in the postsynaptic cell. Very, very rare that a single synapse, a single axon, can activate other cells on its own. So here I activate this cell on its own with this spike that I generate in the soma. It does have EPSPs, of course, in many cells necessary, but most of these cells will not generate a spike. Typically not, unless they are very, very near the threshold for some reason. So most of the key assumption is that there is no loop going backward because this loop, which is going backward, needs another cell here to generate a spike using this spike, unless this axon goes back to yourself. So that we already discussed the possibility that the axon makes a synapse on the same dendrite, on each dendrite, which is very, very rare and very, very small, so it cannot really add, I don't know, 30 millivolts, 40 millivolts, and so on. So after many, many controls and so forth, we decided that this is an action potential that is really going backward, and there are beautiful things. For example, you can block with TTX locally here, you can pass TTX here, so you block the transmission. You don't allow this action potential to go backward because you block locally, and then you block plasticity. So the synapses do not learn anymore because they don't have this signal. There are beautiful, really, really, really beautiful work done on this backpropagating action potential, both experimentally, but of course a lot of theoretical work stick, and many papers on these backpropagating action potentials that I'm not going to talk about. A lot of theories came out of this thing. Okay.
Can you turn the light back on?
Oh, sorry. Okay, so this is like a summary of what I said. Remember this name that you already remember, backpropagating spike or backpropagating action potential. And this is due to this capability, which is really amazing. As I said, nobody in Israel is having the patience to do it. A lot of patience to really implant. Because sometimes you put this electrode and this electrode jumps out, and sometimes you put this electrode and this electrode jumps out. It's not so easy as it looks in the graph.
Where's the red one?
That's why we have models, no?
Okay, now I want to talk about the red one. I want to talk about the red one. Because going even further with these electrodes, another signal was discovered by Matthew Larcombe. I think it was 2002, I think, because they succeeded to record even further in the dendrites of this pyramidal cell. This is layer 5 pyramidal cell, cortical in the mouse. So they recorded here. Note that, as I mentioned before, this action potential is attenuating, although actively, so it's semi-active, semi-passive, as I said, it attenuates, and as you go away, it becomes even smaller. So distal synapses, very distal synapses, will have hard time in knowing that there was a spike, just because it attenuates strongly as it goes backward. So this is the finding of the calcium spike in dendrites. I don't know if you saw this graph. This is another classical by now, sorry, 1999, not 2000. So Matthew Larcombe with Bert Sackman, who was also participating in, Bert Sackman is a Nobel laureate. He was developing the patch technique. It's Neher and Sackman. So Sackman moved to this area, now he's working on the cortical column modeling in silico. We should talk about this, hopefully. So here they succeeded to record it from here, and they did the following experiment, which is now classical. As I said, this is a Nature paper. If you stimulate locally here with your electrode, like simulating an EPSP, so this is the current you inject locally here, and you get a local voltage. Okay, let's say 10 millivolts voltage, the peak voltage. So you generate a local EPSP, so to speak, by your current injection at the electrode. If you only injected the cell body, so this is the black electrode, if you injected the cell body, you get a spike. If it is suprathreshold, then this spike backpropagates, which we know already. So if I record in this location, this is the red spike that reaches this point from generating a spike here. So this is the red spike, and this is the blue spike here, which is bigger, the red spike, and so forth. But if you do simultaneously, stimulating distally here, subthreshold, very locally, and you get this on its own, and at the same time, or actually a little bit before, you activate a spike here, and they coincide, so the backpropagating action potential goes backward and attenuated, plus the local voltage generated here, so this is the case here. A spike at the cell body, a local EPSP distally. Together, you suddenly see a huge spike, This is the red spike. A huge spike generated here. Broad spike. This is the calcium spike. Okay, you see the calcium spike here. Calcium spike meaning that there are channels that are voltage dependent, they are excitable channels, but they carry calcium, not sodium. Calcium. So this is the voltage-dependent calcium channels, like, as I said, the heart action potential, which is based on calcium, not on the ions that flows through the membrane and depolarize the membrane, these calcium ions. This is a calcium spike. So there is this region which locally generated calcium spike. We know that it is not generated here, it is not generated there, it is generated here. So this means that there must be some kind of a local clustering of ion channels that are voltage-dependent calcium ion channels that, if there is enough pressure, enough voltage there, and you could also generate this calcium spike if you activate very strongly locally. So I don't need the backpropagating action potential. I need a lot of voltage here in order to activate this high voltage threshold calcium spike, and then I get this calcium spike. But the interesting thing here is that there is this ping pong effect, because when I get this long, big calcium spike here, I get extra sodium spikes in the soma. So this is the only spike I generated, because I generated the brief current pulse, and I generated one spike at the cell body, plus this injection here. They coincide, and suddenly there is this big phenomena which is now activating extra two spikes. This extra two spikes is coming because there is a huge charge that is generated locally, and this charge attenuates to the cell body and activates the cell body, or the axon, activates the axon with two extra spikes. So this is interesting because there is a ping pong effect. Yes, the action potential goes backward, the sodium action potential goes backward, coincides with local synaptic input, generates big calcium spike, which adds extra, in this case, two extra tak-tak. So if you listen to the cell body, you will hear a burst. Tak-tak, tak-tak-tak. Three spikes.
What is this initial skin in red color?
Where?
Initial skin, it
Here?
goes up, yeah.
Where?
What does this indicate?
So this is the red backpropagating action potential on its own. So this is this one. You see, this is the backpropagating action potential from the black spike to the red spike here. But then, instead of this red spike to attenuate, it is building up. It is building up and generating the whole calcium spike. So this is the backpropagation action potential peak that would have been going down otherwise, but because there is this additional activity here, or excitability here, you generate this big spike. So it's a combination of the backpropagating action potential, which adds local depolarization, plus the local activation that I injected here, and together they succeed to boost the calcium spike locally. So the calcium spike, again, I don't want to go much into the details because this is, again, a whole kind of field, but it's interesting that we did not know that there are two different spikes, at least. We shall show you a third one. Because we are used to think that there is one spike coming out. This is the sodium spike, like in the squid giant axon, there is one spike, sodium spike, that's it. This is what we have in the brain. But I'm showing you that there are other signals in the brain. There is the backpropagating action potential, it's another signal. It's not the axonal spike, it's another signal. There is the calcium spike, again, not in all cells, but in layer 5 pyramidal cells. There is a calcium spike particularly generated in this region. There must be some good reason to have this spike. Yes? They give a very, the whole discussion of this paper is that there is a signature of coincidence detection between inputs that come here, let's say to the basal tree. This is the input that comes to the basal tree simulated by a somatic electrode. There is a coincidence detection of when input is coming here together with input that is coming here. When I'm saying input, I'm meaning excitatory synaptic input. When synaptic input is coming here and synaptic input is coming here, the output tells you that there was a signal, that there was a coincidence. The output detects the output of the burst, tak, 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 detects that there was input here and input there together. That's what they claim. That this particular signature at the output, the signature of coincidence detection of basal input plus top-down input. You know, the input are coming here, are coming from distal parts of the cortex. The input that are coming here are local. So this is a type of computation that the output tells you that there is a burst, meaning that there was this input to the net drive. You can debate on this because, as I said, you can get this burst with a very strong distal input on its own. So again, I don't want to go into it because this is a whole thing that you really should learn. I'm just saying that the biophysics of dendrites, as it becomes more and more clear, the biophysics of dendrites is really complicated. It's not just collecting synapses in a simple way and bringing the synapses to the soma to generate a sodium spike. There is a lot of back interaction, and there is a lot of local boosting, in this case a calcium boosting. And so this is something one has to take into account when he thinks about the brain. You cannot just ignore it, at least in the cortex.
So these kind of action propagation, was it found in other cells as well, or just the pyramidal
it was found also in the pyramidal cells of the hippocampus, the CA1 cells. These are also pyramidal cells. So apparently these very long pyramidal cells somehow want to know that there was a spike. But this sodium spike does not reach here, and you don't want the dendrite to be too excitable, because you can imagine that if the dendrite would become an axon, it would be hard to sum the synapses, because if the action potential flush the dendrite, it really puts the dendrite into inactivation period and so forth. So you want something passive that collects the synapse, but you also want some kind of shaking, handshaking. It's a very kind of interesting solution to be able to sum some synapses still, something the axon cannot do because of this spike flushing all over. But this is a spike that you can, by the way, on top of this passive spike, you can add synapses. You can add synapses on top of the spike because it's not fully spiked. You cannot do it to the spike. The spike is an O09. You cannot make the spike bigger by a synapse. But this spike you can make bigger by a synapse because it's a semi-passive spike. So it's very interesting, but as I said, you really can think about the neuron as a complicated multi-site nonlinearities. It's even more complicated because recently Jackie Schiller in the Technion and Matthew Larcombe found a third type of spike, the NMDA spike. The NMDA spike is really an interesting spike because it's a voltage-dependent phenomena. It's a voltage-excitable phenomena that depends on the transmitter. So the transmitter being released, glutamate, can generate locally interaction between several NMDA receptors, and one receptor, because it is voltage dependent, and another the receptor that also receives glutamate at the same time, and a third receptor that receives glutamate at the same time, together, because of the voltage interaction between them, can amplify the EPSP of each one of them separately and generate something that looks like a spike. This is called an NMDA spike. This was also found in the pyramidal cell. So this is a little summary of a Nature review by Jackie Schiller in the Technion and Larcombe and others, showing that basically in this layer 5 pyramidal cells, and also in the layer in CA1 pyramidal in the hippocampus, also in layer 2-3 cells, layer 2-3 pyramidal cells, also we have this, but not in other cell types. So there is subpopulation of cells that have distal NMDA spikes, more proximal calcium spike, and somatic axonal NA spike. So you can see the three types of spikes.
All the cells you're mentioning, they're all pyramidal, right?
Yes, they are all pyramidal. We don't know of inhibitory cells, for example, having calcium spike, or not yet. It's very hard to record from dendrites of inhibitory cells. So we have still a lot of dark matter. We don't know what's happening there yet. But that's what we know. Telling you about pyramidal cells because that's what we know. And we know that it doesn't happen, for example, as I said, backpropagating action potential does not happen in cerebellar Purkinje cells. But there you do have very strong calcium spikes in the dendrites of Purkinje cells. So, and I'm not going to go anywhere because the whole thing is to talk about computation in dendrites. I really want to show you. So maybe stay a little bit with questions, or maybe finish the question. but not anymore.
Okay, I just wanted to ask if we have channel models that are able to produce these things.
Yeah, of course. We are doing it in our lab. There is model for NMDA receptor. There is, of course, model for calcium. So we can replicate. We can replicate the backpropagating action potential. We can replicate. We have a theoretical paper from our lab replicating this interaction between the sodium spike going backward and the local input generating calcium spike with a burst. This is a work by Itai Chai, which was recently published. So we can replicate most of the phenomena because we have the channel properties mathematically. Hodgkin-Huxley-like models, but for the different types of channels. So I don't want to go more, but there is an interesting theoretical work here. By Bartlett Mell. Bartlett Mell is really the first one who, and I'll show you in a second, his thought about dendrite as a computational device. Bartlett Mell was the first one really to look now to try to really understand the different types of nonlinearities. You know, the different types of nonlinearities, the red nonlinearities, let's call it the NMDA nonlinearities, the blue nonlinearities, which is calcium nonlinear and so forth, in order to put it in a 3 layers of nonlinearity. So looking at the neuron as a three-layer network, but in a very particular structure because it's a dendrite. And so this is one layer, and this is another layer, and this is the output layer, which is the soma. So there are a lot of interesting work, theoretical work coming from this notion. And I just want to summarize, before I go to the computation, just want to summarize for you the main insights that I think I gave you during these past two times and today about the biophysics of dendrites. Biophysics of dendrites. First of all, this, of course, dendrites are electrically distributed, not isopotential devices. There are differences in voltage between different locations and different voltage at different times at different locations because these are cables rather than spheres. A significant portion of the synaptic charge reaches the soma even for distal inputs. Remember, there was this big controversy. Eccles neglected the distal synapses. Roll showed that distal synapses do contribute significant charge to the cell body, although they are very distal. But you cannot throw them away and neglect them. Voltage attenuation and active propagation in dendrites is asymmetrical. Remember the big asymmetry from the input branch towards the soma and from the soma outward, which is more flat. So inherently, because of the boundary conditions, which are different if you go this direction or if you go this direction, the voltage attenuation is asymmetrical in dendrites. And locally, you will get big voltages from the input locally because of the large input resistance in spines or local small branches. Dendrites are significantly time delay. We spoke about that. It will take about the time constant from a very distal synapse to reach the soma. So if the time constant of the cell is 10 or 20 milliseconds, it will take for the peak of the local EPSP to have a peak at the soma on the order of time constant. So this is a significant delay line. That's what I told you last time. This I didn't tell you, but maybe I should not talk about this much. If you look locally, if you want to sum up locally in a small dendrite, okay, so let's say that this is dendrite and this is the soma, and you have two synapses that are impinging locally on a dendrite. I didn't show it to you, but cable theory shows that locally the EPSP will look very brief. Intuitively, it is because most of the current escapes from the input point. So you can generate voltage, but it will be very brief. At the cell body, it will be longer. We discussed the fact that there is a change in shape. There is a broadening of the shape from the input to another location, the soma. So this is much broader. This is on the order of tau. This would be on the order of 0.1 tau. So locally, the EPSP, although it may be big in the spine, it will be very brief because of this current escape outside of this local point. This means that if you want to sum up locally EPSPs, you want to build local voltage, you have to be very exact in timing. It has to be one on top of the other. These two synapses should work very close to each other if you want to build voltage locally. Here, the time window for summation is broader. It doesn't have to be so exact in order for the other EPSP to build up and for the third EPSP to build up. So the time window for integration, or the integration time window of EPSPs is broader at the soma compared to the local time constant for building voltage. Okay? This tells you that if you want to compute, if you want to sum inputs locally, they really have to come synchronously, basically. But if you want to sum them at the cell body, it doesn't matter that this comes here and this comes half a time constant later. This comes quarter of a time constant later. It will in the soma build up well because of this broadening of the EPSP. So that's another biophysical intuition. So this is the temporal resolution that I spoke about. This is the cell body time constant for integration, and this is the order of time constant for integration locally in dendrites. Okay, we didn't speak much about dendritic spines, but this is another. So 7 points to remember. This is in your. This will be in your Moodle. So let's speak about computing the dendrites a little bit, and then I'll give you a break. We start a little bit later, and then we'll continue. If you want a break, if you don't, we can continue continuously, as I said, as we did last time. I want to discuss computing with dendrites. There are a few ideas about what could you do computationally, not biophysically, but computationally. Remember, I spoke about 3 reasons to model. I spoke about, minimal model, key parameters, or explaining experiments, key parameters that capture the essence of your phenomena, and now you want to go to a higher level, which is computing with your biophysical signals. You want to compute with neurons, compute with spikes, compute with synapses. What can you compute? So this is another level of description. Computation, the term computation, which is on its own not clear to anybody. What do you really mean when you say computation? Is it the Turing machine what computation? But we are a computational center, and you have to invite Ron Shagrir to discuss the notion of computation, whether our stomach computes the pressure of the food inside. Do you compute it? What do you compute? Why do we call this machine computer, and the stomach not, or the heart not, or yes? I'm not going to go into this philosophy. So we say computational. We agree that the brain computes something. Okay, so what are the main ideas around this issue of computation in the brain? Of course, there is in terms of neurons, what extra power, so to speak, what extra power does neuron give you because they are distributed compared to a point neuron? So of course, this comes automatically. You say there are subunits locally. This subunit in this part of the dendritic tree, this subunit locally, and the synapses there almost do not affect what happens there because of this distant attenuation. So there could be a lot of interesting local things like plastic events that are disconnected from each other. So it gives you, as we said before, as we showed before by Bartlett Mel, gives you this kind of capability to do local nonlinearities before you sum them up. He calls it pi sigma. You multiply here and you sum up here. Pi sigma computation, if you want. So this is the subunits. I'll show you in a second an idea by Roll and later by others that you can use dendrites to compute the direction of motion. So here I show you, here is the motion. My hand moves from right to left in your case. So you compute this motion. You know that it's from right to left. You know even the velocity. You compute it. So how can you compute it? We can show that a single cell can compute the direction of motion just because it's distributed system. I'll show you in a system. In a second, I'll show you how dendrites can increase the repertoire of firing possibilities, the richness of firing possibilities. If you don't need many channels, just because of this ping-pong effect with dendrites, you can get firing enriched in the repertoire of firing. Bartlett Mel has a wonderful, beautiful paper showing that you can improve if you have many inputs. You can classify them at the output. You can classify which inputs are getting into it because that's what you want. Of course, you get a lot of inputs, and you want to say this set of inputs is different than this set of inputs by your spikes. Your spikes to somehow classify how many classes of inputs do you have. You can do it better if you have dendrites than if you have a cell body in the ceiling or the ceiling. But I'm not going to talk about all of this because, as I said, this is a lot of work and so forth. And I'll show you recent work I just mentioned a recent work by Schiller again, Jackie Schiller and Michael Hauser, showing that nonlinear dendrites, specifically the fact that they are nonlinear, can sharpen the tuning of a cell. Let's say that it's a visual cell and it has this tuning curve. The fact that you have nonlinearity, let's say calcium nonlinearity, can sharpen your tuning and make your tuning more sharp, more exact, more specific. But it all started, of course, thinking about neuron as a computational device started with this dramatic guy. And you heard a lot about, I think you heard about McCulloch and Pitts neurons. So this is McCulloch. He was in MIT. Very impressive, you must admit. And he wrote this book, Embodiments of Mind. It all came from this paper. It is very, very classical and extremely hard to read. And the name of it is wonderful, isn't it? A logical calculus of the ideas immanent in the nervous activity. Rather vague name, interesting name, but a logical calculus. So he claimed that the neuron is a logical element and not, it's a gate. The neuron is an a logical element. You can read it here, but you can read it also at home. But here is the idea, and then I'll finish with McCulloch and Pitt's idea, and we shall continue later after the intermission, because I need to, unless you don't want intermission, as I said. So this is the McCulloch and Pitt's idea. So McCulloch and Pitt, of course, thought about the pond neuron, with no structure, although they knew that there is structure, but they assumed that this is my neuron, and this is the McCulloch and Pitt neuron. And what the computation, or the logical computation, the logical operation that this neuron is doing, for example, just an example, is the following. So these are the excitatory synapses. Let's say that we have 3 excitatory synapses and 1 red inhibitory synapse. And assuming that this 1 red inhibitory synapse, which is not a correct assumption, but let's say that 1 inhibitory synapse can veto completely the output of this cell. Let's assume that the synapse is, the inhibitory synapse is a veto synapse. Of course it's not, it just reduces the excitatory synapse by a certain degree, but let's say that it's a fully veto below threshold. So then I can, and you need three excitatory synapses to reach threshold, let's say. So this will be your logical sentence, your logical statement. E1, sorry, I made a mistake. Each one of the excitatory synapses is enough to generate a spike. So there is an output, either that, or that, or that are active, and not I is active. Then you have, then there is a 1 in the output. Otherwise it's 0, something like that. So this was their idea. Really, in this paper that I mentioned, this interesting name paper from 43, was actually the beginning. I don't know if you are aware of this. This paper was extremely, very, very influential in the computer world, because they gave the mathematics of what you can compute with these and-not gates. In a network of neurons, each one of them is this McCulloch and Pitt neuron connected to each other, and that's what they write here. You can get very, very complicated and-not computation, very, very powerful computation in a network of these interacting elements. Each one is this and-not gate. And so this was very influential paper on computer science, very not influential paper in neuroscience, because people are saying, first of all, there is no veto, and second of all, there are dendrites, and all these kind of things, and third of all, who are these theoreticians who tell us what is the brain is? That we know that the brain is spiked and so forth. So this was a big jump in conceptually, this was the first big jump of trying to connect a cell to computation. Not speaking about the EPSP that is big, that is small, that is before, that is after, spike and not spike, and the mechanism of spike, but giving it a computational meaning. This is a computational meaning to the spike. There is a spike, and this is a computational meaning to the synapses. So this was the first kind of transformation from looking at synapses as voltages, but looking at synapses as computing something. So this was dramatic, but it didn't affect, as I said, biologists did not like this thinking. Today, of course, there is this field that was developed in the 90s that is called computational neuroscience, but it was not a field then. But this is certainly a paper to remember, this 43 paper. Very difficult to read, very, very hard, very mathematical. Very smart paper. And of course, do you want an intermission or not? Do you want a break?
Yeah.
Okay, this time you want a break. So just to finish, this idea of a computational unit that is doing logical operation was extended later, mostly by Koch, Christoph Koch, who is a German working now in Allen Institute, being the scientific director of the Allen Institute. They are doing, as I said, wonderful work in Allen Institute. They already did wonderful work before Christoph came there. You know, the whole brain gene map of mouse and human was done there, very useful for many labs. Now they are doing something else. I hope I'll be able to mention what Allen Institute is doing today. And Christoph Koch is running the scientific aspect of it. And they came with this idea, Christoph Koch and Poggio, who is in MIT, very powerful computational neuroscientist, very, very powerful computational neuroscience, Tommy Poggio. And the idea was there that because you have a structure, a dendrite, unlike you have a point, the location of your synapses become important. So for example, this inhibitory synapse, which is close to the output, so to speak, the spike in the axon, this inhibitory synapse could veto all the excitatory synapses that is in the back of it, that is more distant than it is. These inhibitory synapses can only veto this one. Okay, because when this works, all the current is being shunted here, of this synapse. But of this synapse, the fact that you have somewhere in the back inhibitory synapse is not so important, because most of the current anyway goes this direction. So I don't care that you open a hole in my back. So then you start to write another sentence, another logical sentence, another logical formulation. So it's E3 and not I3, I2, and I1. Or it's E2 and not I2 and I1 because of the location. Okay, so I can complicate here, because if you have really dendritic 3, then of course this one will block all that is coming on this path, and this one will block only this one, and so forth. So the location of inhibition will dictate the logical sentence. So that's a whole paper written by Koch and Poggio based on this idea, extended to the fact that you have cables. So this is really the first time that people assign computational meaning to synapses and spikes. But we shall continue after a little break. Let's say 10 minutes break or so.
היה איזה משהו שצייר את הפולינום של זה עכשיו, טיפ 2. כן, היה לך פולינום, אני זוכר? כן, הגדרתי לך שזה אומר שיש כאילו שורה, יש כאילו 0, היה פה אחד שיש 1, אחד שהיה 2, נכון? אבל מה זאת אומרת? למה מילה אנסטייבל?
מה למדה חיובי על זה?
אם הוא חיובי, זה הלמדה? נכון, זה הלמדה.
זה הלמדה שבוע,
אז אם הלמדה חיובי, אז זה אומר שיש לי אייגן ואליו לפחות אחד שהוא חיובי?
חיובי זה אם יש לי נכון, אם יש בוודאות זה אנסטייבל. אם הוא החיתוך מחשב שבכיוון אחד זה סטייבל, אז צריך שבשלושתם הוא יהיה סטייבל כדי שיהיה...
ואיך אתה יודע?
לא יודע. אם זה חיובי, אני יודע שבוודאות...
אנסטייבל. למה אבל?
יש לי כיוון אחד שהוא אנסטייבל.
מה זה כיוון שהוא אנסטייבל?
יש איזה אייגן וקטור שמתאים לאייגן ואליו, אז בכיוון של האייגן ואליו, אם האייגן וקטור זה אייגן ואליו הזה, אז הוא מתבדר, לא מתחמם. אני יודע שזה אנסטייבל.
למה הוא מתבדר?
אמרנו, חיובי זה בורח, זוכר... שמתי בגרף, שזה שלילי, אז אני כאילו... אתה זוכר להגיד בסטלן נוד, אז אחד חיובי ואחד שלילי. אז כאילו נשם אנסטייבל.
לא יודע, אתם מדברים עכשיו על שאלה 2, סעיף C, אתה צריך לסווג את הביטקוין? כן? למה אתם מצחקים בשביל שאלה?
לא, אני פשוט חייב לאכול את זה.
לא ראיתי שאתם...
לא, אני.
הוא עם הסנדוויץ' אז הוא יכול...
כן, נו כן.
אז זה רק לפי, יש לך בעיקרון, אם אתה חושב את הפירוט, יש לך 3 נאמות, אחת שהיא ממשית, ושניים האחרים, שתיים אחרות הן
זה אומר שהלמדה שלה,
מצאתי למדה אחת לכל פיקס פוינט.
סבבה, אבל כולם חיוביים, כל הלמדות חיוביות.
כן, אבל לא מצאתי את הלמדה אחרת של אותה פיקס פוינט.
יש למדה אחת.
לא, יש עוד שתי למדות שהן לא ממשיות.
בסדר, לא ממשיות, זה לא מעניין אותך.
כן. כי אז אתה אומר שיש לך כאילו שלושה קומפלקסים, שאחד מהם שלו הוא אפס, ואחד מהם הוא לא אפס.
לא, אבל זה לא מעניין אותך, כי ברגע שזה אנסטייבל, ברגע שאחד הוא גדול מאפס, ולמדה אחת גדול מאפס, זה אומר שכל הפיקס פוינט הזה זה אנסטייבל. כי מספיק לך אחד, מספיק לך למדה אחד כדי שתתבדר.
ראיתי שהיא כתבה את זה בסביבות הזאת בשביל השבוע, אבל למה?
כי, אני לא יודע אם זה סבבה, אבל...
לא מישהו כתבה את זה, לא הבנתי למה.
כי כשאני אשאל ככה, מה זה הלמדה? זה מתאים לאייגנווקטור, נכון? מה זה אומר? כאילו, תחשבי את זה בשני אייגנווקטור. עכשיו, אם אחד מהם הוא גדול מהם, ואחד קטן מאפס, אז אחד נכנס פנימה, נכון? כדי שזה יהיה סטייבל, כדי שיהיה סטייבל, כל האייגנווקטור צריכים להיות שליליים. כדי שזה ייכנס פנימה. כי שלילי זה לתוך הנקודה.
אייגנווקטור?
האייגנווקטור, כן, האייגנווקטור שלילי, ואז האייגנווקטור נכנסים כאילו פנימה.
ואז זה סטייבל.
כן. אבל אם אחד מהם הוא הולך החוצה,
you always get, if you don't get this one, it means that there is no cycle at all.
In case of calcium,
Yes.
if we
Oh, in case of calcium.
if we get only this technique and without this one, so we say okay, this is population of neuron or this area from where I'm imaging
Where
is not
active.
active.
Okay, let me see that we understand each other. You see, if there is a somatic action potential, if there was for some reason, maybe input comes here, let's say, or there is enough synapses coming here, if there is a somatic action potential, it's always back propagate to the dendrite.
Okay.
Always.
Okay.
So it goes from here to here and will generate locally this spike, it will be this spike, actually this here. So this is only on its own.
Yeah.
It will be this spike here, it will be this spike here. So the red spike in the dendrite is a signature that there was a sodium spike in there.
Okay.
Also.
I'm relating this thing with the calcium relating thing in which we inject some calcium dye and we record the calcium signal.
So you will see here, by the way, you see this little
yeah, we
bend.
do see this kind of long signal.
This is already calcium.
Yeah.
So you see that the backpopulating action potential in this case already activated a little bit of this calcium, but it's very local, so this is a hump.
Right.
It's a local hump.
But like when we say okay, whether this is a response or this is not a response, if we get this case, then we say okay, this is a response.
Response from a spike going backward. Yes. If there is a lot of calcium locally from imaging, huge amount of calcium, it probably must mean that there are either a big calcium spike, this one which really brings a lot of calcium, so this would be flushed with calcium, a lot of calcium due to the calcium spike.
This is
a long and broad and high, so there is a lot of calcium going into the membrane. This will be very, very, very full of calcium because of the calcium spike. But the confusion can also come because when you have these NMDA spikes, which is not here by the way, it's more distant, this will also of course bring calcium locally through the NMDA receptor.
yeah.
So this is another source of calcium going in.
How do you differentiate between NMDA and this is calcium?
Because in NMDA you can block the NMDA, there is a specific blocker. Q, whatever the name is, I don't remember now. There is a specific block of the synapse. So you don't allow the magnesium to go away. So it's blocked. So then you don't get calcium through this. If you continue to get calcium, it must mean that it comes from channels that are voltage gated, not transmitter gated.
Calcium. So in the previous slide, the calcium signal that we are looking at, it may be the combination of NMDA and all the calcium.
Exactly. Could be a combination of that.
And the second.
But you know when in the very first that I showed you that Arthur Konos is doing 2-photon microscope very, very locally in the spine spine itself, we know today that the membrane of the spine itself has NMDA receptors, so calcium can flow through this when the synapse is active. If the synapse succeeds to reach a certain voltage, because of the AMPA, then the NMDA receptors will open and you get calcium. But also on the spine, there are calcium-dependent voltage channels, and this will another source. So you don't know here. You don't know here you don't know here if you see calcium, whether this calcium comes from NMDA or from the other one or both. This you don't know. But you do know that this synapse is active for this direction and not for this direction or this direction. So you see the calcium in this direction, let's say, so this synapse is sensitive to this direction, and this synapse is not sensitive to this direction. So the calcium, not knowing about the origin of the calcium, is the fact that the synapse works and there was voltage here. This voltage may open NMDA, the same voltage may open
the calcium. Okay, and the second thing, like the non-linear and dendrite. So we do say that there is neurons are non-linear.
in the output. The output is highly non-linear.
So
Because there is
this is the resultant of all the like non-linearity in dendrites result in the non-linearity of neurons.
Exactly. That's what this is what I meant to show you about this maze. So there is non-linearities in this dendrites because of NMDA. So it's a local non-linearity because of this mechanism of NMDA spike. There is non-linearity that is more central. Mostly here. Maybe because let's say this is the calcium non-linearity here. Maybe here. It's not clear exactly who is assigned to who, but there are local non-linearities. There is another local big signal here, and then there is summation, so to speak, or multiplication of all these non-linearities, because it is summation, although this is of course very non-linear as well because of the spike. So there are these three sources of interacting non-linearity. It is non-linear here, summing here, generating non-linear here, summing there, generating non-linear. So these are the three layers that it speaks about, so to speak.
And third question is like, the more the non-linearity, the more it is tuned.
That's what we showed.
So
That's the paper that we showed.
what is the meaning of getting tuned? For example, auditory cortex, if you say is tuned to this frequency, this neuron is...
Let's say that the neuron is tuned to this frequency, and some of the amplification to this frequency comes from nonlinear channels. Let's say that I block this channel only. Then the tuning, the boosting, the difference between this angle, let's say, or this frequency to this frequency is less dramatic. The nonlinearity, if there is a strong nonlinearity, in the preferred direction there is additional boosting, so the response will be big to this direction, or to this frequency. To another frequency there is less nonlinearity, so the nonlinearity may just exaggerate the separation between the preferred and the non-preferred. It's just a local mechanism that can really boost everything that is in the correct direction or the best frequency. And in other frequencies it is much less activated. So it's a boosting mechanism to sharpen, to make it more different between this frequency and this frequency. That's the idea of people, and they show this experimentally.
Which paper?
These are this.
I want you.
This is Hauser. This is in the 747. This one.
Okay.
Schiller in Nature Notice 212 showed that for a whisker system, it was a visual input. She recorded from V1 cells in the mouse, and she characterized the receptive field, the tuning curve of this particular cell. Then she blocked, if I remember now, I think she blocked NMDA, not NMDA. She blocked the calcium channels, and she showed that it becomes wider. It's less charged, the tuning curve. The same is done also by Hauser in this paper. So these are the two papers that show the dendritic nonlinearity, specifically dendritic, sharpen the tuning curve of the output.
Okay.
So it's some help in the computation, because if you want to sharpen, to make the cell very tuned, you want to reduce the sides and to increase the center. So you want to make it more sharp. You you can do it with local dendritic nonlinearity. That's what they show. So tell me a little bit about you. Where are you from? You are part of MTech now?
Yeah.
Are you doing a PhD or just
Yeah,
one year?
no,
You're
I'm
doing part.
doing PhD.
Where did you come from originally? I mean, India.
Yeah,
But
Delhi. And I did master's physics,
where?
Delhi University.
I see.
And then I spent like 1.5 years in the Neuroscience Institute, and I was
Where
yeah, in
in New
India.
Delhi?
And I was working in this auditory neuroscience space, mostly with computational stuff. And now I
So
am
you came
working.
directly from there to here?
Yeah.
Wow. It's a big jump, huh? It's a
I'm
cultural shock.
yeah, I'm in LS lab now, and I'm doing this culture embedding. So I'm interested in
I see. So you're thinking about culture in terms of
yeah.
yeah, but Ellie worked a lot with Connor.
Yeah,
And they have a wonderful paper
I
together.
both came from yeah,
Oh, I see.
and some
Oh, so that's how
yeah,
you
he
know them.
taught about the calcium
I
matrix.
see, I see.
And this
Wonderful.
is interesting stuff.
I think it should be relevant to what you do. That's why
And
we teach you all these things, you know, that
yeah,
eventually one day
and
it
now
may affect your thinking.
initially people like most of the things we did in computation, the spike count and rate, it is with the action potential. Now where do we use it? Dendrite experiments. Now I see these are
Well,
the more
but this is
important.
very difficult technologically. You really have to have this. I don't exactly know what succeed. What did Connor succeeded to do there that nobody else is doing to push the two photon microscope to the level of synapses. It's really rare, but he succeeded to do it. And that's why everybody came to work with him. Ellie went to work with him on the auditory system and Tuckman on the whisker system because he could really record locally from there.
Do you think that we can switch from the spike count of action potential to the spike count of dendrite and do the
Yeah, but limited because it's only few locations for now.
a particular
A
network?
particular network, local. But then you have to really see about what is the unique thing that they succeeded to do technically, which I don't know really. I don't know what is it. It's a lot of optics to push the signal to noise
So
eventually.
it is in the paper of 2010 of
Yes.
okay.
Oh yeah, there is a whole big, you know, the discussion about how do you do, which I never read personally so deep because it's not my
We
field.
can invite him to give us a
Of course,
contribution.
he comes a lot to Israel. He's a friend of Yossi Aron, Connor. It's very good. And he's friend of everybody now. Let's start because it's time. Okay, let's continue because okay. Okay, so let us start. It's always surprising to me because I feel that I have so little material to tell you, and it comes that I never succeed to finish it. This time I asked for 3 times. Usually I gave two lessons in one presentation. By the way, I found that my presentation, that your presentation on this work, the papers that you will present on this work is on the 3rd of May, so not this week only. So this I solved, and I also asked Adi how to upload the lectures to Moodle, so it should happen in the next few days. He's not in Israel, so he should write to me how to do it. Anyway, so I want to continue as much as we can to discuss the computational aspect of dendrite, and I want to show you what does it mean a directional selective neuron, in this case in the retina, So without saying too much, because I'm sure you know that you can give, in this case it's a ganglion cell in the retina, so it's an isolated retina. You take the retina out of the system, reverse so to speak, and you put light in this direction, or grids in this direction, this direction, this direction, and you see that this particular cell that is impaled intracellularly give rise to EPSPs in all directions, but to spikes in these two particular directions, 225 and so on. And then you can draw these plots. You can draw the plots, the directional selectivity plots for the EPSPs. So you see the EPSPs becoming bigger as you approach the preferred direction. And then of course the spikes are more narrow because you get spikes only in these two angles. So this is the orientation selectivity of the spikes and so forth. So you can see directional selective set. And so the original idea that came from Roll 64 was that a single cell can do this computation, provided that it's being, as I said, mapped, as I will show you, mapped correctly synaptically. So the basic idea is the following. So this is the 64 paper, and this is a model of a neuron. This is the cell body, and this is the dendrites, compartments of the dendrites. And in this case, in this particular model, all the dendrites, all the compartments receive excitatory synaptic inputs. So these black circles are excitatory synaptic inputs. And you can activate these inputs in two temporal orders. You can do A, B, C, D. So you activate this excitatory synapses stop, then this stop, stop, stop. In this direction, in time. So first proximal, more distal, or the same synapses but reverse order in time. You start distally, more proximally, more proximally. So it's either A to D or D to A. It's the same synapses, it's just reverse order in time. And you look at the EPSP, the composite EPSP, it's not a single EPSP because it comes from synapses, from distributed synapses. You look at the EPSP, the cell body, and you see different EPSP. If it's A, B, C, D, you see the first EPSP coming from this. The first EPSP coming from the nearby synapses. And then the second one, which is more distal, it takes some more time to build up on top of the shoulder. So this EPSP on its own would do something like that, the nearby EPSP. But then this one was activated as well. So this one was first activated, this one second activated, and so forth. They start to build one on the shoulder of the other. So this is temporal summation of EPSPs, one on top of the other, like here. So the first one wants to decay, but then there is another EPSP coming distally, and the third one wants to decay, there is another coming distally. So you get a broad EPSP, but a relatively lower amplitude EPSP than in the reverse direction. What happens in the reverse direction? You start with the distal one. It takes time, of course, to reach the soma. But on its way to the soma, it sums up with this one, on top of its head, so to speak. So this EPSP comes here, some with this EPSP here. The two of them come here, some on top of each other here, and so forth. But it takes time to reach because it starts distally. So you see that it takes more time to reach, but then you build bigger. And if I had a threshold here for spike in the output, you can see that one will not give me an output, and this will give me an output. So the output will be directional selective, assuming that there is some particular mapping between the world and these synapses. So it should look something like that. So I drew it for you graphically. Suppose this is your ganglion cells in the retina, or any directional selective cell. This is the world, the visual world outside of us. And this is a point moving in the world, so this is this point moving here, or this point moving there, okay? And these synapses are mapping the world systematically onto the dendrites. So this location in the world is mapped here, so there should be some very specific mapping between the world and the dendrites, systematically, if this was the case. Then this would become a directional selective cell just on the basis of the biophysics that I just showed you. Okay, because if you move from here to here, you will get something like the blue one, and if you move from here to here, you will get something like the red one, and and you get an output in this direction, so this will be the preferred direction of the cell, and this will be the null direction of the cell. Of course, it will depend on the velocity of this point, because it has to match the delays of the synapses. You can imagine that if this was very fast, if it will sweep very fast, you will psychophysically not see the point moving, but also the cell will not see the output, also this direction. So of course there should be some matching between the physics of the input, the physics of the optical input, to the physics of the dendrites in terms of delay. People like myself and others showed that this particular system is relevant to the way we see speed moving or point moving in the world. But this is a direct computation done with synapses and dendrites and spikes based on mapping, particular mapping, and the fact that you have delays in dendrites. So this is the origin of the difference here, of course. The origin of the difference between these two temporal sequences is the fact that there is a delay in the dendrites. Of course, if all the synapses were in the cell body, if there was only a point neuron, you cannot do this computation. There is no meaning to first, second, or second, first. It's all in the same location. But the fact that you have a structure gives you a capability to do a specific computation, directional selectivity here. And you can, of course, extend it. So how do I know that I move my finger from here to here, or from here to here? Somewhere in the center, somewhere, all these receptors should sum up. And the output of this somewhere should be different than this and here. Otherwise, how do I know that I move here, there, or there? And this is the same in frequencies. How do you know that I do the somewhere there should be something that sums up the difference. So this is directional selective in the general sense. I just gave you an optical example. That there should be some point that sums up and knows, so to speak. Of course, there are many ideas how to do it with networks. The dynamic of the network will be different in different orders of inputs. I'm not saying that single cells are doing it. I'm just saying that you could do it with single cells. And that's, of course, the problem of this field in general, of computational neuroscience, because we have very nice ideas that comes from very clever people from physics and so on that are coming to the field. But there is very, very rarely a direct proof that the brain is really doing this computation, both at the level of single cells, and at the level of network. There is very, very rare examples that the theory met the experiment in a one-to-one way. Of course, these ideas are wonderful because then you do experiments to try to validate or refute these ideas. They are very important, but that's the problem of the field, that we didn't find one concrete, clear, proof-full example that this is how the brain computes. So I'm giving you an example of a possible computation. Okay, but I don't know that it works like this. But people work very hard on trying to show that indeed there are these ideas, and one of these key ideas is the one that I show you now, and this comes from the fly visual system. Again, a German group, what is called the Reichardt detector. So Reichardt, who died recently, was a very wonderful and very special person, was the supervisor of Axel Borst, who is now in Munich, that we work with in our group on the fly visual system. And this Reichardt had this idea that you can compute on this cell the direction of motion based on a formalism that I showed you one example of, how to implement this formalism, whereby you have a line with some delay. This could be dendritic delay, it could be synaptic delay, it could be network delay because there could be another cell here. Some asymmetry, some origin of asymmetry. Could be dendritic asymmetry, synaptic asymmetry, network asymmetry. Some asymmetry will cause the output here to be sensitive, so this is supposed to be the receptors of your point. So this is the Reichardt receptor. This is the Reichardt detector. So suppose you start here, you have a point of light, let's say, moving here. There is a delay line here, and they meet, because of the delay line, they meet here, and they generate an output. In the other direction, if this would have been the first, then there would be no coincidence in time, because this will come first, this will come later, and plus a delay, They will not sum here, they will not multiply, so to speak, they will not sum, and there will be no output. So this is the most simple, the most basic elementary computational device because of this asymmetry that you embed into the, if you want the network, if you want the neuron, if you want the synapses, I don't care now, but in principle. So people are looking for the Reichardt detector. Physically, they are looking for the Reichardt detector. And you know,
the Reichardt detector in this...
So this is the Reichardt, this is the Reichardt detector.
Oh, okay.
This is the Reichardt detector. The idea that you have asymmetry due to some time delay without specifying what is the origin of this time delay. As I said, it could be dendritic delay. I showed you before an example, but it could be some other source of delay. I'm not specifying anything. This is a general conceptual, it's a conceptual thing that you break the symmetry of these things by having some delay line. That's all, and then you sum up at this location.
So can the dendritic spikes be the delay? I mean,
that will be originated only in one.
Yeah, because maybe the first photoreceptor caused an action potential, but then the dendritic spikes cause a few more spikes that continue to...
Exactly. But not in the other direction. For some reason, this direction of activating synapses, let's say this is your dendrite, in this direction there will be no spikes, or in this direction, or this direction there will be spikes, or in this direction for some reason there will be no spike. You have to have some asymmetry in the lines. They cannot behave exactly the same relative to this one, but but if there will be a dendritic spike that is slow because of some reason in this direction, and that is not slow in this direction, then it will be implementing Reichardt idea. I just want to show you that people are looking for it really, really hard by using this. So there are other ideas, but all are based on this correlation based. You have to correlate this input with this input. There are other ideas how to implement in the end neuron, in the computational neuron, the Reichardt detector idea. The most fundamental idea in direction selectivity. And so people are trying to really see whether you can find such a thing, let's say in the network in the retina. Because I know that in the retina there is this ganglion cells that are directional selective. I know it. I showed it to you. But what is the mechanism to make it directional selective? What is the mechanism? Is it inhibition in one direction and excitation in the other direction? This could be also, you know, there are many other ideas. So this is just a general, I just want to show you that there are these ideas, and that people like Mitya Kishlovsky, who was in Janelia Farm, now in New York, is really doing this hard work. Hard work, because when I said, for example, that you have to map the synapses in the dendrite idea, synapses have to be mapped from the receptor, then you have to show this mapping. So you have to do this EM work in order to show that there is a direct mapping, let's say, in this origin. So this is what is called the connectomics. I'm sure you heard about this term. This is the world effort. Whoever has a very powerful EM equipment to reconstruct pieces of dendrites or pieces of networks or pieces of neurons to find out the location of the synapse and say whether this is excitatory synapse, inhibitory synapse, and really start to build the network from EM slices. There are several groups in the world. The leader used to be really an inventor of the two-photon microscope. Another German, you know, Germany, you cannot compete with them. They are fantastic in science, in many fields. They are very good. You know, I just mentioned many of them just in passing, not that I plan to do it. Axel Boss and Reichardt, and of course Wilfried Denk. So Wilfried Denk is the guy who developed the two-photon microscope, and he developed a new method to do the connectomics, whereby it takes a block. Unlike what they showed here, you saw that when they do this block, there are many ways to do EM. You can take a slice here, EM slice, put it aside, EM slice, put it aside, and so on, and then image this one, image this one, image this one, image this one after the other. And then there is a problem of alignment. You have to put all the images aligned so that you build the correct network. So Wilfried Denk developed this other technique that you take the whole block and you scan it from above, and only then you slice. So it's already aligned. You don't need to align it, but you scan it and then you throw this part, this piece, this slice. You scan it, you throw this slice. So he's doing a wonderful work, Wilfried Denk, who is now moving to Munich from Heidelberg. And and he recently came with a very interesting paper. This work. So this is now anatomy. This is EM connectomics anatomy. Yes, this is really hard work. So this is the ganglion cell, the directional selective cell. So if I will record from this cell, as I showed you before, it will be directional cell, and it will prefer this direction. So there will be spikes in this direction and no spikes in this direction, or less spikes in this direction. And he found something interesting anatomically, an anatomical finding, because of these slices, and reconstructing the network, that this is an amacrine cell, and this is inhibitory amacrine cell. So this is a cell, amacrine cell, that makes, this is a presynaptic cell to the ganglion cell. This is the third layer in the ganglion, in the retina. The axon that is coming from here is the axon that is going to the thalamus. So this is the output of the retina. The optic nerve. The optic nerve, these million axons are coming from million ganglion cells in our retina. So this is the end, this is the directional selective cell that we recorded from. This is one cell before. So this is a presynaptic cell. This is a very interesting cell, this amacrine cell, because it doesn't have an axon, and the output is from its own dendrites. So it's a dendritic synapse. From dendrite of amacrine to dendrite of ganglion, and this is inhibitory synapses, and you can see the asymmetry that you need for Wi-Fi detector, because the asymmetry is that on this side, on the right side, there are more red than on the left side, as you can see. So if I move my point from right to left here, I first activate the amacrine cell. This is presynaptic cell, so it's first activated in time. So I move it here, it's activated, and it puts a lot of inhibition on this cell, because there is a lot of inhibitory synapses. So there will be no output if I go this direction, but if I go this direction, there is less inhibition coming from this side, less inhibition, less red point, and then I will activate this cell by another, the bipolar cells, and there will be an output. So this is the first time that somebody, by the connectomic method, showing the asymmetry in connectivity built into the network. It's built in, so to speak, to the network, in this case, to the ganglion cell, which explains the directional selectivity. Again, we can discuss it a lot, whether this is enough to explain or not. I'm just saying that this is an effort, because we understand that something about the mapping of synapses within the network, or at a given cell, the neuron level, dendrites and so on, is important to understand the computation. And this is an attempt to do it, the work of, as I said before, you may know, maybe, I don't know if you know this work, like Shklovsky, and the whole Janilia farm is dedicated to the eye, to the visual system of the fly. They work a lot on the fly system. It has about 800,000 neurons. They want to reconstruct the whole brain of the fly at the EM level, and then explain the capability of the flies to do all the things that it can do based from the anatomy to the computation. It's very debatable, and people like Chaim Sopolinsky, for example, do not like this direction. It's not like physics, this is too detailed. Of course, you would say rightly that there was first a theoretical idea. The theoretical idea came from a theory, the Reichardt theory, and then you're looking for something. Otherwise, you will look until the end, but you don't have an idea, so what are you looking for? So, of course, it should come together. But maybe this is too detailed or not too detailed and so forth. There is a huge controversy because it's a lot of money also, the project. The connectomic is extremely expensive. So maybe we throw money or not throw money to get a map of the brain at the synaptic level. Maybe we can do it at the light microscope level to get some principles statistically, not needing every detail and every synapse. These synapses are plastic sometimes, so maybe today you have this synapse, tomorrow you have another synapse. So it's a statistic of synapses. And I don't want to go into this connectomic debate, but it's a big debate and a big project. Because you have the tools. You can cut, and reconstruct. Not in Israel again. No connectomics work in Israel. Okay, so the other issue, and I'll go fast because I want to mention also some other things. The other idea that I mentioned in this list of possible computational things that dendrites can do, you can show that if you have, so this is work of Mel, of Rinzel, and this is the work of Grinzel also showed it, that if you have a passive dendrites and active soma, so this is the somatic spikes, the Hodgkin active spike, and the dendrite would have been only passive, and you inject current to the soma, you will get, let's say, a regular spiking activity. But if you have an active dendrite, like a calcium spike, for example, that I showed you, and there is this ping-pong effect between this nonlinearity, which is the sodium nonlinearity, to, let's say, the calcium nonlinearity, you can start to get an interesting output behavior just because of this ping-pong. You can get a bursting activity because of this ping-pong. And the claim is that if you want to get this behavior from one compartment, you have to really work hard in terms of which channels do you put exactly in order to get a burst. So this is an inherently a clock, so to speak, that goes from a spike here to a big delayed spike here, which goes back here, and so forth. So just the fact that you have more than one compartment and a nonlinearity and interaction between them can give you a rich repertoire of firing.
Why do you have a gap between the two?
Because it takes time for the spike here to generate the calcium spike here, because of the slow dynamics of the calcium spike here, let's say 100 milliseconds. So you go and there is the back propagating action potential, which takes another few milliseconds, and until it comes here and goes back here, this would be the gap of this ping-pong effect. So it's a matter of the kinetics, the dynamics of the channel.
But shouldn't it have at least as many spikes during the time window as the earlier one? Yeah, shouldn't it? Because why shouldn't you have those intermediate spikes like you do in the previous case?
Why do you have what?
In the first case with the passive dendrite, you have spikes the whole time. Here you have spikes in the beginning and spikes in the end. So what happened in the middle?
In the middle. So again, I don't want to go into the kinetics of these channels, but these particular channels that they put in the model, it's a particular model that you can read in the paper. This channel have also another type of channel. It's called calcium-dependent potassium channel. You know, there are these interesting channels that are open when you have a big calcium event. You open voltage-dependent potassium channels that shut off, like also the regular spike. After the spike, the sodium spike, you have a potassium that blocks the spike. But there are these very slow A current, it's called. And this A current is slow, and if you activate the A current, it blocks the capability to fire a spike for a long time. It's like a long refractory period due to this A current. So again, I don't want to go into the mechanism. You can read this paper, but you can get an interesting firing because you have ping-pong effect between two dynamical systems. It's like an oscillator, two oscillators, but there are different types of oscillators here. This one is slow, this one is fast, and so which are channel oscillators. And again, I don't want to go more into it, but I want to finish because we don't have time. I just want to show you one of the big projects that is very debatable. And because I'm part of it, I want to tell you that not only me, but also Sackman is another part. So there are these two attempts that you may know. One is the Blue Brain Project, that became now the Human Brain Project, the European Human Brain Project, which is very controversial. There was a lot of debate whether it's a good direction to go or not. And there is the group of Sackman, Oberlander, and what's his name? I forgot now the name of the third guy. Moritz Helmstatter, who became a Max Planck director in Frankfurt, doing this in silico cortical column. So what is the idea here? You really want to take a full network now and try to understand the dynamics of this specific network, really specific network. In the case of Whisker, it's a column, it's a barrel cortex. So it's a really particular network in the barrel system. It's a very, very specific network. In the visual system, for example, there are